Good morning and welcome to the Forest Growers Research Conference for 2021. My name is Grant Dodson and I have the pleasure of introducing the second session on day one titled Industrial Forest and Wood Products. For those of you who don't know me, I'm based in Dunedin where I work at City Forest Limited as CEO. I also contribute to the industry through a number of roles with Forest Owners Association, Southern Wood Council, and I sit on the Forest Growers Research Board. Following my short introduction, today's session will be 5 15-minute presentations, followed by a 20-minute Q&A session, and we hope to conclude at 12:05 p.m. So, running you through the presentations. Presentation 1 begins at 10:30, and it'll be brought to us by Steve Wakeland from Scion, who will present on the Tree Root Microbiome Program, which is a transformative platform for forestry. Steve is a senior scientist at Scion, specialising in forest ecological genomics and tree-soil interactions in a changing environment. He has a PhD in plant pathology and experience working for the CSIRO in Australia as an environmental microbiologist. Before joining Scion in Rotorua, Steve worked at AgResearch as a soil and plant microbiologist. Steve's presentation this morning on the Tree Root Microbiome Program will firstly touch on innovation in forestry science and the balance between applied research that will make a difference today alongside the need for the discovery of new areas of science that can create value for the industry in 10 or more years' time. Steve will then introduce an exciting new joint MBIE/Forest Growers Levy Trust funded program that is aiming to develop the next generation of tools for our sector to rapidly adapt to a changing climate, as well as the potential discovery of new value opportunities from wood and forest products. Presentation 2 at 10:45 a.m. will be brought to us by Simeon Smale, who will talk on the Resilience Forests Program, which is a portfolio of opportunities for raising forest sector productivity and reducing risk. Simeon Smale is a senior scientist at Scion, specialising in plant-soil microbe interactions at various scales. He completed a Master of Science exploring nitrogen cycling in red beech forest during Masters, and followed that up with a PhD investigating the long-term impacts of radiata pine site management on soil microbial community activity. Simeon joined Scion in 2006 as part of the field team and has subsequently made his way up through the organisation in various roles and positions to his current role as senior scientist. This morning, Simeon will provide an overview of the Resilient Forest Program, touching on the foundations of the program and what it is designed to deliver for the New Zealand forestry sector. Simeon will also describe the research areas that underpin the program and provide details from a range of projects illustrating the depth and range of the research that the Resilient Forests Program enables. Presentation 3 at 11 is from Cherie Belvett, the Nutrient Science Manager at Balance Agri-Nutrients. Cherie's presentation will focus on the Biuret product and will cover a quick recap of the potential of Biuret, why Balance is interested in developing Biuret, the activities and research being conducted in order to bring this product to market, and finally, the timeline for delivery. Presentation 4 at 11:15 will be brought to us by Stu Fraser, who will present on the epidemiology and control of red needlecast. Stu is a research group leader in the ecology and environmental section based at Scion. The aim of this research is to develop recommendations for cost-effective chemical treatment of red needlecast. The approach that Scion have taken around red needlecast is to undertake a series of in-field disease monitoring and controlled lab studies to understand the epidemiology of the disease, which is all about understanding where, when, and why disease outbreaks occur. This information can then be applied to help determine when control operations should be applied within a year, in which years control measures are necessary, and in which areas control measures are needed. Stu will then talk about the large-scale field trials that aim to test the efficacy of copper in controlling red needlecast and optimal spray timings. We'll also hear about the work undertaken monitoring the disease impacts on tree growth, which then feeds back into a full cost benefit analysis of the control operations. Presentation 5 at 11:30 a.m. is from Helen Whelan, Lincoln University, who will update us on progress with trichoderma in New Zealand forestry. Helen has a PhD in plant pathology from Lincoln and has undertaken disease research in horticultural crops at Hort Research and the Plant Protection Research Unit at Lincoln. Helen then became a winemaker, a manager and owner of 4 vineyards in Canterbury and Marlborough, and also a grapevine propagation nursery. She's also been involved with property development. Helen returned to Lincoln in 2017 to extend her research capabilities through postdoctoral fellow and research officer positions in the Bioprotection Research Centre. Following the retirement of Dr. Robert Hill, the lead researcher of the trichoderma research program, Helen took over the role of research into beneficial trichoderma in New Zealand forestry. Today's presentation is focused on utilising the power of trichoderma root endophytes for forestry to enhance bioprotection and production. The talk outlines a process whereby trichodermas were bioprospected and selected for growth and foliar disease suppression in radiata pine and other forestry species. This promising work is currently at the stage of validating trichoderma blends and ultimately aims to commercialise a trichoderma bioprotectant for improved growth and tree health. So the final part of today's session will begin at 11:45, and that'll be a simple 20-minute question and answer session, and that'll conclude at 12:05. Thank you, and I hope you enjoy the presentations.
Good morning, everybody. Tēnā koutou katoa. My name's Steve Wakeland. I'm based here in Otatahi, Christchurch with Scion Research, and I'll be talking to you today about the Tree Root Microbiome Program, which is an innovation hub for the future of the forestry sector. So the Tree Root Microbiome Program is an exciting new innovation space which is really set up for tomorrow's forests. So as I said before, my name's Steve Wakeland and I'm presenting on behalf of the Tree Root Microbiome Program team. So the problem statement that we face is that in the ground today, growing across all of New Zealand, are planted forests worth around $60 billion. So the forestry sector is sitting on a massive asset base. And we all know that the sector is somewhat unique in that it takes a relatively long time to realise returns. So roughly 3 decades sit between the day that stock is planted, you go out in the field and you plant your stem, and then when you can go and collect your wood and get your return on your investment. This inevitably means that there's an extended exposure to risk, whether that's from pests or diseases, weather or climate, or even national shifts in policy, policy direction, or access to markets. Of any sector, forestry has arguably the most highest exposure to risk, but also the brightest future if we can get it right. I mean, there's been no greater need for forests and forest products, and this need is growing. Benjamin Franklin famously said, "Change is the only constant in life. One's ability to adapt to these changes will determine your success." Given the pace and breadth of change in the forestry sector, it's clear more than ever that we need to be investing in new ideas that will allow forests and forestry not only to survive, but to thrive into the future. If the sector is going to adapt to risks, capture opportunities, we must have invention of new ideas and space and opportunity to develop and explore new ways of forestry. The Tree Root Microbiome Program is an innovation engine room. It's there to provide the conditions for invention to occur. But before we go into what the Tree Root Microbiome Program will be doing, let's quickly touch on what is creativity and what is the creative process. So creativity and invention is all about connecting dots, connecting things in entirely new ways. It's what happens when you're tinkering away in the back shed, in your art studio, your writing room, or your other creative space. You might, in fact, deliberately go into that space just to escape from other distractions and allow creativity to happen. It's the very start of the pathway that leads to new outcomes, whether for the forestry sector or the IT sector, doesn't matter. It's the start of the process. And without a stream of new ideas, innovation eventually dries up. Sectors that don't innovate stagnate. They're not able and agile to change. Typically, the most transformative and impactful outcomes come from unexpected, unlikely, and disparate connections of ideas. A good example is some tinkering from a group of scientists at the Radio Physics Lab at CSIRO, Australia. They were investigating black hole astronomy and particularly methods for signal correction. Now, it's very, very hard to get clear signals coming from black holes that are millions of light years away into your recorder in a room in Canberra, Australia. There's a lot of signal interference and bouncing around and things to consider. They paired up with some groups that were looking at brand new methods for signal correction and married these with some new mathematics based around Fourier transformation. It's hard to imagine, looking back, how they put these ideas together, but looking into this problem, when they connected the disciplines around signal and error correction and Fourier transformed algorithms, in doing so, they inadvertently solved a huge issue that was emerging for the IT sector at the time. They essentially invented Wi-Fi. That is, distortion correction for wireless networking. This is now used in about 5 billion devices all around the world and came out of the Black Hole Astronomy Lab tinkering around working out how to do signal correction through deep space before the samples got to their laboratory. So creativity and invention require quite specific environmental conditions to occur. And there's a really good understanding around what these are. So let's just try and understand these for a moment. So let's ask the screen legend, John Cleese, maybe one of the most creative people that has been in the last couple of generations, his thoughts on creativity and what makes creativity. Since retiring from the comedy stage, John Cleese has often been called upon by corporations such as Google and others to share his knowledge around creativity and the conditions needed to foster it. He draws from his own experience in his creative life, but also on the research conducted by Professor of Psychology Don McKinnon and others that was conducted in the 60s, 70s and 80s. First of all, it's really, really important that creativity is not a talent. It's a way of operating. Creative people have acquired a facility that gets them to switch into a way of operating that allows to open the creative part of their mind, to enter their shed or their art room. Professor McKinnon describes this as the ability and time for the mind to play with ideas and explore them, not for any immediate practical purpose, but just for its own sake. To get your brain from the closed and open and creative state, there are some key conditions, and these conditions can be created. The first one is space. Space away from usual pressures and demands. Nothing dampens creativity like interruptions, distractions, and constantly having to do things. The second one is time, setting boundaries around the space, a beginning and an end, providing an oasis that separates you from the doing mode, the day-to-day mode, but rather invests in sitting with and exploring problems from different angles for longer periods. And the final condition is confidence. Nothing stops creativity more than fear of making a mistake. The very essence of play is being open to whatever happens. If you think any direction could be wrong, you can't be in a creative mind. So the Tree Root Microbiome Program is a space that has been deliberately made with specific time and resource boundaries to support and foster creative thinking on the role of the microbiome in tree and forest fitness. It was created with a very clear view of the need for the forestry sector to think and develop a base of new ideas and innovation needed to progress and support the sector for the next 20, 30, 50 years' time. It is there as an innovation space, providing opportunity for the sector to be agile, to develop new opportunities for shifting forest phenotype, increasing fitness, controlling disease, and maybe even altering wood properties. In short, if we don't provide the space for entirely new areas of thinking, innovation will stagnate. Changes imposed on the sector will outstrip our ability to respond or change our toolbox of options. What we know already is that microbiomes play or profoundly alter the systems that they inhabit. We know this from the human microbiome model. Often these outcomes are completely unpredictable and very transformative. We have a very strong evidence base already from previous investment in the forestry sector, the growing confidence in forestry futures and healthy trees, healthy futures programs show that the same is true for microbiomes in trees and forests as they are in humans and other model systems. And globally, there is massive transformation potential for harnessing the microbiome. The question is, what are the creative, innovation, transformational opportunities for the tree microbiome program? So the tree microbiome program is around connecting the dots in new ways. An example will be connecting our knowledge around the radiata pine genome and our resources associated with genetic variants and variants trials across New Zealand and Australia. We're going to connect these with advances in analytical microbiome communities, both in or on plant tissue. And the image above shows a portable handheld DNA sequencer that plugs in via USB into a laptop. We're going to connect our plant genome information and the genotypes with all the microbial populations in entirely new combinations, in entirely new ways. We're going to explore the outcomes of these interactions on the plant phenotype at the Plant Accelerator Phenomics facility in Adelaide. Essentially, we're going to ask, can we breed new phenotypes into Pinus radiata via the microbiome? Can we use these combinations, these new dots, these new ideas as ways of generating new tools for disease resistance, drought tolerance, climate adaptation? We are connecting the plants and the microbes in new ways, and we don't really know what to expect that the outcomes of this might be. We need to expect the unexpected here, and that is where the confidence part of creativity comes in, being open to whatever emerges from this work. Another example is connecting the hypothesis that Darwin put forward in the 1800s, that the root of the plant acts like a brain and has a role well beyond anchoring the plant and sucking up water and nutrition. We're going to test this by eavesdropping on the chemical conversation that's happening between the plant root, the microbiome, and the metabolome. Furthermore, we're going to model our approach on the human gut-brain axis, which tells us that the gut microbiome is in reciprocal communication with the brain and work together to affect the overall fitness of the host. And finally, we're going to be doing all this at the New Zealand Biotron facility based at Lincoln University, where we can use laparoscopes to go down into the soil and actually understand in real time all of these interactions that are happening on the root microbiome soil surface, and start to really understanding all of the changes and all the signals and communications that's happening between the plant, the microbiome, and the environment. And we aim to answer these questions. Are roots cognitive? Can we show a pine forest how to adapt to change in the future that it hasn't seen before in the past? And finally, we're going to take advances in ecological DNA analysis, connecting those with advances in environmental typing, providing smart apps and standard operating procedures to laboratories all around the world to take up and use and go out in the field and measure Pinus radiata, its genomics in the environment, and a whole range of settings that New Zealand will eventually move into with a range of climate change conditions. We aim to set Pinus radiata up as a global model pine microbiome system for everybody to use. By engaging the global scientific community with us on this process, we will empower the adoption of the model and the de facto standard for tree microbiome research globally. We just imagine how powerful it would be for New Zealand's forestry sector if we had labs internationally all focusing on Pinus radiata, growth and productivity, and all developing new tools for manipulation and assessment for New Zealand. It would have incredible value. So what will the forestry sector outcome be? Ideally something that we're not even prepared for yet. A discovery or discoveries that will enable entirely new and transformative opportunities for the sector. The outcome's going to be a sector that is more resilient and robust and agile to a changing future. And in short, the sun has never shone so strongly on the forestry sector. The demand for forests and forest products is growing rapidly. There's lots of opportunities for the sector to innovate, to change, and to realise these. And the Tree Root Microbiome Program is proud to be on part of this journey with you. That's the end of my presentation. I thank you all for your attention and look forward to questions at the end of the session.
Good morning, my name is Simeon Smale from Scion, and I will be presenting information from the Resilient Forests Program in this presentation. Although I will be presenting these set of slides, there's a huge team that underpins the activity in the Resilient Forests Program, so I just want to acknowledge that wide and diverse contribution as well. So, the establishment and the goals of the Resilient Forest Program is where I'd like to start. So the program itself builds on past research that was undertaken in the Growing Confidence in Forestry's Future program and the Healthy Trees, Healthy Future program. So selected research areas and concepts from within these two past bodies of work were identified and discussed with a range of stakeholders, including representatives from the Forest Owners Association, Forest Growers Research and the New Zealand Farm Forestry Association. And that research was discussed and iterated to form what became the basis of the Resilient Forests Research Program. And one of the key concepts underneath that was considering what would happen if we followed business as usual practices from now into the future and what the impact that would be on our profit when we consider an increasing body of risks that the industry is facing, such as loss of social licence, poor forest health, potential for reduced wood quality, culminating in a significant potential for productivity decline. And some projections were done that show that there's a potential for an annual loss of $1.3 billion in net present value, taken from projections from 2018 values. So the Resilient Forest Program is really looking to address these issues, and indeed the work that we've done showing how what could happen if we mitigate these factors or indeed advance beyond them shows that if we can gain public endorsement, greater support for forestry, if we can improve forest health, if we can produce better wood quality, overall resulting in enhanced productivity, there is a potential to actually go the other direction and not just save $1.3 billion that could be lost, but actually add a further $1.3 billion in annual net present value. So overall, the idea is to use the research around enhancing productivity and improving public endorsement to de-risk current and future forest investment. Now, the Resilient Forest Program is based on three central research areas. The first one of these is managing uncertainty and risk. Now, this involves components that are around looking at things like applying portfolio approaches to assess multiple risks simultaneously, things like changes from climate change, market issues, disease incursions and public concerns, and then using that information to develop tools and frameworks to help anticipate where those threats might occur and what the potential responses to them might be that maintain productivity and social license to operate. And then thinking about how you can actually apply those processes within adaptive management for our forests, so they are future-proofed against whatever risk might come along. The activities that are going to be undertaken to deliver on these goals include looking at the drivers of social licence and really trying to understand what people actually want from forestry and how perceptions of forestry and how forestry is managed will shape future investment. And putting that all together, there's another activity around understanding how we can use that information to actually help manage risk, both physical and social, while retaining that social licence. So there's a lot of work that's undertaken under these areas. Some of that's going to be reported in detail in future presentations, but I'm just going to provide a couple of snapshots from some of the work done under RA1 now. So looking at the triggers and drivers of social licence. This was explored with a survey followed by focus group decisions, taking in a wide sort of swathe of people involved in forestry, one way or the other. And overall, across a diverse range of communities, it was found that forestry was valued and forests and forestry practices were generally considered acceptable. However, there were sensitive issues and that were identified around the need to consider environmental benefits, aesthetics and other personal values and how those were reflected in forest management. And there was also a very clear signal that the New Zealand public does want to see investment from forestry into native forests and alternative management tools. But it was also really clear that they didn't expect the entire forestry sector to change to these. It's just if there was some investment that would be seen as beneficial and it would reflect well across the entire forestry sector, whether it's still conventional forestry or using some of these alternative management tools. Another area that we've looked at is how we grow our forests and the implications of that. So there's a lot of work from other primary sectors looking at the wider impacts of land management and crop management with regard to is this activity of benefit or a risk to social, environmental or cultural values, just going beyond financial and economic returns. And it's become an increasingly important factor in market access and consumer decisions. So to look at this for forestry, Sion engaged with a panel of experts who are involved in looking at trends in primary sector industries, and a number of key points came out. That FSC certification was going to be of increasing significance. Reduced chemical use was clearly going to be considered advantageous for market access, and there was a need for a clear chain of custody. This piece of wood was provided by people following good labour policies, and that the soil and water resources that built that piece of wood are protected. And this goes with the trend of increasingly seeing investment determined by environmental, social and ethical criteria. So next I'm going to move on to the next research area, which is productivity enhancement. And this contains a range of various topics which I'm sure are familiar to people, such as improving nutrition, improving phenotyping and modelling tools, looking at the role of the microbiome, then moving on to the silvicultural side, looking at opportunities to improve stand uniformity and productivity. And then if all of these things work and we have faster growing, better trees, what are the impacts of shorter rotations on wood quality? And again, the activities going on under this area really map onto these well. So again, looking at changes in wood properties, modelling tree growth and dynamics, also maximising knowledge gains from our long-term trials such as Puraki and the accelerator trials, enhancing our remote sensing capability, looking at responses to nutrient management and exploring the role of the microbiome. Now again, you'll be getting some presentations on specific aspects of this work, so I'm just going to provide an overview of a couple of areas here. So in terms of nutrient management, we're looking in detail at nutrient responses. This is being looked at in the accelerator trials, driven by the use of the nutrient balance model NUBARM, really figuring out the best possible moments to add nutrients, and nitrogen in this case, and trying to get as much as possible in that into the tree to get the biggest possible growth gain. And that's actually working really well at those sites where we're getting some strong successes there. But we're also going beyond that a little bit, trying to improve precision in predicting fertiliser response, not just looking at the pools of nutrients at a site, but also understanding how much of that pool is actually available. So it's changing the paradigm a little bit, but it is proving to be quite beneficial. Another area that we're working in is looking at micronutrients. So manganese, molybdenum, zinc, for example, as well as boron. Now, we're not just taking the approach of trying to diagnose what micronutrients the current crop of tree needs. We're actually looking at how we can apply it to the site so we get the entire ecosystem out of a micronutrient deficiency. So not just the current crop will be more productive, but all future crops of trees at that site as well. And we're also trying to underpin this work by improving our ability to analyse nutrients through high throughput systems. And these are based on spectral analysis. So the first area that we're looking at is using remote hyperspectral analysis. So basically flying a drone over a forest, and that is able to take readings with hyperspectral imagery of the nitrogen and phosphorus in the needles of the tree. So we can sort of get this real-time map of the dynamics of the nitrogen and phosphorus through the forest. And then in the lab, we're using infrared analysis to predict soil properties, a much faster and more efficient approach than traditional chemistry-based analysis. Another area of note is the work being done in the microbiome of needles and wood. So we've developed a new protocol to detect oomycetes, probably better known to which include Phytophthora in radiata pine, and we're looking to deploy this across further trials as well. And we're also making quite significant advances looking in the microbiome present in the bark and wood. So we've got samples now that show that rather than being sort of a desert, we're seeing a diverse microbial community living inside the cambium and the wood of the trees, that are quite old in some cases. So we're looking at potentially up to 70 in fungal species and 400 bacterial species living inside the tree. So this raises a lot of questions around what are they doing and what are our opportunities to influence them, because if those fungi are present, if those bacteria are present, they are active, they are doing something. And this could potentially provide significant new opportunities to alter the tree metabolism and the wood properties within the tree after it's actually been formed. The next research area that the program is supporting is looking at enhanced resilience. And this includes things such as the development of alternative disease control tools, looking at the epidemiology and spread of needle diseases, tools for surveilling disease emergence and movement, and also some work looking at the actual impacts of disease. What's the relative impact of changes in the extent of disease on tree growth and wood quality through time? And again, the activities that are being undertaken in these areas map pretty closely to these goals. So again, epidemiology and spread, the landscape level effects of disease emergence, opportunities to create resistance to pathogens through potentially greater diversity of the tree crop, but also chemical control and disease management. So a couple of overviews from this area include looking at epidemiology and spread. So Scion has purchased some high-resolution imagery over a wide area of the New Zealand planted estate. And we're looking to use this to help improve a predictive model that can provide early indications of disease severity. And this imagery is actually able to be processed and pick up the impact or the incidence of disease prior to the naked eye being able to pick it up. So we're working with an external provider, Indafor, to develop a method to automatically identify red needlecast outbreaks based on this imagery, far before we'd ever be able to see it in person. There's also work going on using UAV captured imagery to score individual trees to understand how the extent of the disease that they might have, red needlecast in this case, is actually going to impact their growth through time. So again, important information to understand the resources we need to commit to fighting red needlecast. Another area we're working in is looking at alternatives to copper. And recently there was a workshop at Scion held with scientists and stakeholders, and that really discussed stakeholder perspectives on the future of copper in New Zealand. How long can we keep using it? And what information do we have around alternatives to copper, different control tools that we could use? And the next steps from this workshop is to develop a roadmap for operational tools that could be used against Dothostroma in the short, medium and long term in a world where copper is not available. So another area I'd like to touch on is the interactions between these research areas as well, which is a real strength of the program, that we have research going on in quite distinct areas, but we can pull them together within the structure of the program. So if you consider the intersection between RA1 and RA2, there's clear overlap in terms of looking at shorter rotations through enhanced productivity, but what does this mean for market risk? What does this mean for wood quality and where those products might be able to be sold? Between RA1 and RA3, again, there's clear interaction space between thinking about how we can integrate disease risk and climate change risk simultaneously. but also thinking about how our options for chemical use, current or in future, how they might be viewed by the public and what social risk elements they might bring. But there are also huge overlaps as well between RA2 and RA3 when you consider the potential for crossover between pathogen and between the management of pathogens and beneficial microbes. How changes in genetics to improve productivity, changes in genetics to improve in silviculture to improve productivity might enhance disease risk, and integrating disease and biotic risk into growth models, so again we can understand how disruptions to growth should actually properly be modelled. And put all together, this just adds to the platform we're trying to build around de-risking forest investment. Well, thank you very much for your attention. And at this point, I'd like to acknowledge all the hard work and effort that's gone into this program from all the researchers that are involved in it so far. But I'd also like to acknowledge the efforts of the forestry companies who have supported this work and the access they have provided to the various trial sites that underpin the work we are doing. And I'd like to take any questions on this presentation at the end of the session. And thanks again.
Hello everybody, thank you for coming along to hear my talk. My name is Cherie Balvert and I'm with Balance Agri-Nutrients, and I'm going to talk to you about Biuret from concept to product development. Now, I know that many of you are familiar with Biuret. You've probably heard a lot about it in this very forum in recent years. In relevant applications for Biuret that are relevant to New Zealand Pinus radiata forestry industry in particular were tested under the Growing Confidence in Forestry's Futures program. Essentially, Biuret is a source of nitrogen that conifers do well on. Other plants, however, seem to display some very negative effects, or what we call phytotoxic effects. This is where the plant growth is impacted severely, sometimes to the point of death, and sometimes from just very, very low amounts of Biuret applied. For conifers though, and pines in particular, we see some very positive effects on growth, and these growth responses are over and above what we are seeing with the same amount of nitrogen applied in a different form. As well, we are seeing positive effects on the nutrient uptake efficiency of nutrients other than nitrogen. Particularly phosphorus and boron, if they are applied alongside Biuret, we see a much greater uptake efficiency of those nutrients. Biuret appears to have a positive effect on soil microbial function and activity. So the soil microbial pool is being stimulated in some way, and it may even be primed for release of nutrients from the organic nutrient pool. There has also been some evidence of an increased tolerance to disease with plants that have received Biuret. Perhaps the increased plant health and vigour means the overall resistance to disease, if there is an outbreak, is improved. We also have evidence that seedlings grown with Biuret as a nitrogen source outperform stock that was treated with conventional fertilisers when planted out in the field. And what I think is perhaps potentially the most exciting application of this product is if we can manipulate or utilise, as I discussed earlier, that sensitivity of some plants to Biuret. And this could help or result in a reduction in herbicide applications for weed management if the application is timed appropriately. I have environmentally friendly option there in the question mark, because theoretically, although yes, yet to be proven, this could be an environmentally friendly option for improving production in Pinus radiata forestry. Biuret is significantly less water soluble than nitrogen in other forms such as urea or DAP, so it should have a much lower potential for nitrogen leaching or nitrogen loss via ammonia volatilization. As well, if we can have a reduction in herbicide or even fungicide use in these systems, that is obviously also an environmental win. So why balance? Biuret is essentially a plant nutrient or a fertiliser, and this is what Balance does. We manufacture and we distribute fertiliser for our customers. This is our core business, and so this is quite a natural business fit as we see it. As well, Balance owns a urea manufacturing plant in Kapuni in Taranaki, and Biuret is produced through a process similar to that used to make urea. As well, this Kapuni plant is the only urea manufacturing plant in New Zealand, and it is a relatively small plant, which means it is quite agile and can be manipulated to include biourea manufacture. Balance has a supply chain that has a national footprint. We have the ability to distribute the product throughout the country. As well, we also have an aerial application branch to our company. This is called Super Air, and this will assist with application of biourea to some of those more remote forestry areas. Balance has an environmental strategy that we are calling Balance with Nature. Through this, we are doing our best to assist our customers to maximise production on their land with the lowest possible footprint. One of the ways we do this is by ensuring we are supplying the most appropriate and best product to enable this. We know that nutrient addition and fertiliser addition in forestry is increasing for increasing production or perhaps shortening rotation lengths in those stands. And it is becoming apparent that biourea is probably the best product that we should be providing. Less a why Balance, but I just wanted to take the opportunity to point out that Balance has embarked on a Sustainable Farming and Fibre Futures program in partnership with Ministry for Primary Industries. This program has a $25 million R&D spend commitment over the 5 years of the program, and biourea is a big part of this program. It is not the only project, but it is a significant part. This program has a 60-40 funding split, with Balance paying 60% and an MPI 40%. So where are we at? As I said earlier, a lot of the effects or the benefits of biourea were tested under that GCFF program. However, that program used a product that was 99% or greater biourea. We know that we cannot create a product on a commercial scale that will have a biourea content that is that high. So we have done a bit of work to understand what the minimum biourea requirement that product must have to ensure we are also observing those benefits. As well, we're cognizant of the fact that that program was largely carried out on a single soil type and in a single region in New Zealand. So we have expanded that research to ensure we are seeing a consistent response across all the different soil types. And next we will be moving into different regions to cover off different climates as well. We would also like to understand what impact biourea applied early on has over the full life cycle of the stand. We understand it is a long-term investment and we want to ensure that that is safe. Now that work is all very important, but definitely the vast majority of our time and energy and spend at this point is going into these following points. One, the mode of action. At this point, we do not understand how or why we are seeing this response of pine trees to biourea, and also why we see a starkly different response with other trees. It is really important that we understand how biourea works to ensure that we will see a consistent response when used in the field and used widely, but also to ensure we won't see any unintended or unintentional consequences, particularly of a negative nature, when this product is more widely used. Environmental and safety impacts, quite obvious why we need to look into these. We have, however, conducted a very in-depth toxicology review of the product, and we are confident that there are minimal safety or toxicological concerns with its use. However, we are required to provide evidence to this point. So we are carrying out some both soil and aquatic toxicology and ecotoxicology studies. As well, it's important that we understand the impact on non-target species. What are the consequences if this product were to wind up on food-producing cropland or farmland or native biodiversity areas, for example? As well, we'd like to look into some of those theoretical environmental gains from the use of biourea over other nitrogenous fertilisers. Regulatory approval, all this information and all this data that I've mentioned above will be required for us to gain approval to manufacture and sell biourea within New Zealand. However, as well, the regulatory process in itself is quite lengthy and complex. So we are putting our effort in now to ensure that those ducks are lined up when we do have product available to sell. The engineering puzzle, and this has been a puzzle. Theoretically, it's quite easy to make biuret. You heat urea until you have biuret. However, on a larger scale, it is quite difficult to have the control of the reaction to ensure that the end product is what was desired and intended. So we've literally gone back to the drawing board, and this here is a picture of a back of an envelope drawing from one of our engineers, and I think they've had quite a lot of fun figuring out how to play this one. And we have created successfully a product on a lab benchtop scale, and we now have design for which we are going through a business case to build a pilot scale plant. The pilot scale plant will give us the confidence to scale up to a full manufacturing scale plant following that. I wanted to give you a bit of an understanding of the timeline that we are looking at. Assuming you are all chomping at the bit to receive this product, I wanted you to know how long you will be waiting. We are looking at a 5 to 8 year program to deliver this product to the forestry industry. We are currently in concept development, and we plan to initiate building of the pilot plant that I just discussed in next year, 2022. This plant will only have the capacity to produce about 500 metric tonnes of product per year. We realise that will only service about 2,000 hectares of that. However, this will allow for operational scale field trials to ensure we are seeing the benefits of the product. As well, as I said, it will give us confidence in the manufacturing technology to allow us to invest in a commercial scale plant. We anticipate producing biuret on a commercial scale around 2027. This is essentially my final slide, but I just wanted to take a couple of minutes to talk through the key drivers of our business case and bust a few myths, if you will, around biuret. Biuret is not a byproduct of our urea manufacturing process, and we do not have a stockpile of biuret looking for a home. Biuret is a contaminant in our urea product. However, over the years, the manufacturing process has been streamlined, if you will, to the point that biuret is significantly less than 1% of our urea product now. This means that specialised capital and equipment will be needed to be invested in order to manufacture biuret. We will be taking high quality liquid urea from our plant as the feedstock to the biuret part of the plant. That urea already has a market, and we can sell that today. However, and we then also need to further process that urea to create biuret. This means that biuret will be more expensive than urea because it simply costs us more to make. As well, that urea that we are using up here, we need to replace, buy elsewhere and import to service our current market. But biuret has additional benefits also. This plant, we believe, will be a world first. It has positives and negatives. It is very exciting to be a part of this. In the fertiliser industry, it's not often we get truly new products. However, there are negatives. We are going through the trial and error ourselves, and we don't have any ability to import to seed or prove the market, if you would, before we are required to make a significant capital investment in manufacturing capability. I'm not sure if you noticed in the previous slide when I said commercial production, we are still only looking at about 8,000 metric tonnes per year. And if we have gauged the size of the market correctly, we do not believe that that will service the entire market. But that is for now, and we may be able to scale that up in the future. As well, we believe that it is a large biuret market. However, it's largely a few of you out there that are just large players. And this is quite different to how we normally operate, and it is perceived as quite a risk for our business case. So you may have had us out knocking on doors or someone on our behalf just to understand what are the key drivers for this product. So to conclude, Bioret is an exciting opportunity for the forestry industry and for Balance. In Balance, we believe Balance is the natural owner for this product. It will be a long journey, but we are well on the way and we are excited to be doing it alongside you. Thank you for your time, and I'm happy to take any questions.
Good morning, Atamarie. My name's Stuart Fraser and I'm a forest pathologist and research group leader at Scion. Great. So today I'm going to be talking to you about epidemiology and control of red needle cast. So the overview of the talk. First, I'll give an introduction to red needle cast for those people who aren't so familiar with the disease, and just a recap for those who are. I'll then outline the research aims and approach that we've been taking. I'll talk about research we've been doing looking at the epidemiology of the disease. So this is all about understanding when, where, and why epidemics occur. I'll then go on to talk about some of the copper control trials we've been running, building on earlier work that demonstrated that copper had promise in controlled lab trials. So we've taken that further into field trials. And then I'll talk a bit about some work we've been doing looking at growth impacts of disease. And then I'll wrap up with some conclusions. So just as a bit of an introduction, red needle cast is a disease caused by two species of Phytophthora, which are fungal-like microorganisms. They're not fungi, but they act a little bit like fungi. Phytophthora pluvialis is the main cause of the disease, but sometimes we also get another species out called Phytophthora canovii. This disease was first discovered on the east coast of the North Island in 2008, but has likely been present in New Zealand for a longer period. This disease can cause significant growth loss after defoliation events. So in one study, it was found that 38% of growth was lost the year following a cast event. So this disease can have a significant impact on growth. So the aims of this work are to develop recommendations for cost-effective chemical treatment of red needle cast. The outcome being that industry can manage red needle cast in existing stands now and into the future. The benefit being increased productivity, at-risk stands are protected, and the industry retains its social license to operate. So the approach we've been taking is to undertake research to better understand the epidemiology of the disease. So that's the where, the why, and the when disease outbreaks occur. So we're doing research monitoring disease expression year on year to understand why we get outbreaks in certain years and not others. We're doing work within years, looking at when spores are released and when infection occurs within a year. That will give us a clue as to when it's best to apply treatments. And we're also undertaking controlled studies to parameterize an epidemiological model, which can kind of help us get into the black box that is red needle cast, understand the different processes better in the disease cycle to be able to predict when outbreaks may occur. The other area we've been working on is looking at copper as a potential control tool. So copper is used widely for control of Dophystroma, and previous work had shown that it showed promise in controlling red needle cast also in controlled lab and glasshouse trials. So we've taken that further into the field, so I'll talk more about those trials. And we've also been doing some work trying to understand the growth impacts of disease so as to enable a cost-benefit analysis. So I'm going to start off talking about the epidemiology of the disease, so the what, the where, the when, and the why disease epidemics occur. So the first thing to understand is that red needle cast is a very seasonal disease. So this graph shows monthly detections of Phytophthora pluvialis in our diagnostics lab over the last 12 years. And what you can clearly see is that we get peaks of disease in the middle of the year, which is indicated by these red bars, which show the winter months. Conversely, in the summer months, which are indicated by these green bars, the free summer months, we get very few detections. So I'm just about to show a video of red needle cast expressing in a year, this year, over on the east coast of the North Island. So this video starts at the end of May this year and runs until August, the start of August, just before lockdown. And if you kind of concentrate on the fourth tree to the right, you'll see at the start of the video there's the red needle cast is also already expressing there. But as you'll see as this video runs, how quickly this disease progresses under the right conditions over winter. So as you can see, it started with a small amount of infection in one tree, and it quickly spread up the tree and to neighbouring trees, causing a large amount of cast by midwinter. But it's important to note that timing within a year can vary. So here we have a study looking at detection of inoculum at a site also in the east coast of the North Island over 3 years. And you'll see in the first year we had peaks in detection in autumn and spring, but in the second year we didn't get a peak until early summer, and then finally in the third year we got a peak in midwinter. And another really important thing to note is the repeated heavy outbreaks year on year are rare. So here in 2008 we have a stand that was really heavily affected, but then in 2009 is green and healthy. So work we're doing this year and under the Resilient Forests programme is to develop a epidemiological model which will help us understand and predict when an epidemic of red needle cast may begin, peak and end based on forecasted weather, and to enable us to forecast whether a disease epidemic will occur in a certain year. So this model separates needles into susceptible, so that's the S on the left, and these are uninfected susceptible needles, and as the epidemic progresses they move to infected but still green needles, and then transition to infected red needles and then infected cast needles. And I'll just demonstrate this with some photos now. So you'll see in this photo there's a little bit of red infection in the middle, and then most of the needles around that infection are probably green and infected needles. As the disease progresses they change to infected and red needles, until finally they're infected and cast needles. So to help parameterize this model, this year we've been doing work looking at the impact of temperature on different life stages of the pathogen. So you'll see on the top 3 graphs, we have 3 different time points after infection, and you'll see the top 3 are sporulation at different temperatures. The bottom 3 show symptomology development at different temperatures. And what you'll see is we had no spore production and no symptom development at 25 degrees. We had a very little amount at 5 degrees, but we had pretty much the same amount of sporulation at 10, 15 and 20 degrees. What you'll note though is although we got the similar amount of sporulation at 10 degrees as at the warmer temperatures, we didn't get very much symptom development. So this indicates, and we see this at the higher temperatures as well, that we get sporulation before symptom development. So this is a bit like, potentially a bit like the COVID super spreaders who don't show symptoms but are able to transmit the virus. So in these trees that we see that look green and healthy but transition to red very quickly, could this be partly explaining it, that there's infection there that isn't showing symptoms? I'm now going to talk about some of the copper trials that we've been doing at Kinleaf the last few years in close collaboration with Mike Baker of Hancock Forest Management. So in all trials, we've used the Dophystroma standard copper formulation in our trials. So in 2017, we had one application in February and an unsprayed control, and we, as I said, applied at the Dophystroma standard. And what we found in this year was that disease was significantly reduced in sprayed blocks compared to unsprayed blocks. We followed this trial up in 2018, but as well as having the February spray time, we also included other spray timings. So we had a November and a May spray timing. And what you'll see, we had it at 2 sites. You'll see on the graph on the right, we had no difference between the treatments. But on the graph on the left, you'll see the spray treatments, which are in green, had lower, significantly lower disease severity than the non-spray treatments, which are shown in red on the left. So again, copper applied at the Dophystroma standard reduced severity, but we didn't see a difference between those timings. We then followed this up in 2019, again looking at the different spray timings, so including a November, February and April spray. And what we found was that the November and February spray significantly reduced disease severity, but the April spray did not. What I must say though, in the 2018 and 2019 years, there wasn't very much disease in the trial, so we can't make any sweeping conclusions yet. Ideally, these trials would be replicated under greater disease pressure. So now I'll just talk about a few follow-up experiments where we're trying to look at growth impacts of the disease. So we've kind of extended these copper trials, again at Kinleaf, working with Mike Baker of Hancock Forest Management. And we have different spray timings. So we've got a February spray, an April spray, and we've got unsprayed blocks. And importantly, we've incorporated permanent sample plots, PSPs, into these blocks. So we can try and exclude disease using copper and then look at the impact that the disease is having. So this trial has been running since 2019 and is planned to run for at least 2 or 3 more years. So far, disease has been low in the trial, so I don't have data to present, but this year disease has been more significant, so hopefully I'll have data to show in due course. So I'll just wrap up now with some conclusions. Within the Resilient Forest Programme, we're working to develop an epidemiological model which will help us predict outbreak years and support management decisions for controlling red needlecast. Copper applied at the standard rate for Dophystroma control is effective for controlling red needle cast in the trials so far, but the disease levels haven't been significant. So ideally we would replicate these trials under higher disease levels to see if it was still remained effective. Again, we didn't see any consistent effect of spray timing, but again, we need to replicate this under greater disease pressure to be sure. Optimal spray timing may vary across regions and between years, so that's where the epidemiological model can come in and help us understand those processes and that variation. And work is underway to quantify growth impacts of the disease, which will allow a full cost-benefit analysis of control operations. So I'd just like to acknowledge Mike Baker and Hancock Forest Management and Dukin New Zealand for helping with this research, allowing access to the forests and collaborating with us. And then lots of people at Scion have been involved in the research, and there's just a list of some of them, so thanks to them as well. And thanks to you for listening, and I would be happy to take any questions.
Ata Marie, good morning. I'm Helen Whelan of the Bioprotection Research Centre at Lincoln University, and I'm here today to give an update on the trichoderma project in New Zealand forestry. The objective of this project is to develop a long-term symbiotic relationship between the forestry trees and beneficial trichoderma root endophytes. So we're looking to improve the economic returns by increasing production, and this may lead to shorter rotation times. We're looking to help alleviate the cost of current and future foliar disease epidemics, particularly when we have more impacts of climate change. Trichoderma can also offer a non-chemical product to help reduce the agrochemical use in nurseries and forests. Now, this project is mainly concentrated on radiata pine, although there has been some work done in Douglas fir and cypresses. So I'd just quickly like to review what trichoderma fungal root endophytes are. They're a naturally occurring fungi found in plant roots and in the soil, and they have a symbiotic and mutualistic relationship with plants, so both are getting benefits. So we've collected hundreds of strains from the roots of very healthy pines, natives, cultivated and weed plants throughout New Zealand, and we've tested them for growth benefits and disease suppression, mainly Dothostroma, terminal crook, and Diplodia in greenhouse and plantation trials. I'd just like to point out that trichoderma does a similar job to mycorrhiza fungi. They can both improve nutrient uptake, promote plant growth, and reduce the effects of environmental stress. But trichoderma is particularly good at activating the plant's defense systems, and they can be aggressive against pathogens. So the main difference between trichoderma and mycorrhiza is that trichoderma is not visible to the eye, and it's often inside the root, compared to mycorrhiza fungi, which are usually seen outside the root. Now, trichoderma fungi, they're relatively easy to propagate and they're easy to use in the nursery, with the most cost-effective, practical and efficient application method being as part of a seed coat recipe, although they can be applied as a soil drench with commercial equipment or as a root dip. So there's been good uptake in 2021 with 10 nurseries using inoculum in their production. An important question we have looked at is how tolerant are trichoderma inoculants to commonly used agrochemicals and other products. So this is information for nurseries that want to continue using agrochemicals. So we did a large study testing 14 fungicides, 2 fumigants, 14 herbicides, 6 insecticides, 4 fertilizers, 5 adjuvants, and 6 adherents, biostimulants, and a bird repellent. So we tested the strains that made up the PR6 and PR3A blends, these being the final 2 blends that were selected throughout the project. Now, these agrochemicals were generally recommended for use in forestry nurseries and plantations, and they were applied at recommended label rates, mainly for nursery use, although a few of the agrochemicals were applied at the higher plantation rates. And if you would like to get the details of the study, it can be found in this report on the FGR website. Now, there were 2 types of experiments done. The first was in the lab, where the agrochemicals were applied to the spores, first in solution. So we were looking at whether there was an impact on spore germination. Also, they were mixed in with agar to see if the growth of the fungal mycelium was affected, and we looked at the sporulation as well. Now, these experiments were considered direct contact between the trichoderma and the agrochemicals, and this gives information on whether spores can be mixed with agrochemicals in spray tanks if you choose to do this. The second set of experiments were 8 greenhouse experiments using either soil or potting mix. The agrochemicals were applied as one application. They were applied at different times, depending on the type of agrochemical. We also did a seed coat experiment. Now, in all cases, the seeds were inoculated with trichoderma at sowing, and obviously the trichoderma grew into the roots over time. Now, we considered these greenhouse experiments to have indirect contact between the trichoderma and the agrochemicals, apart from the seed coat experiment. And over time, we measured the levels of trichoderma in the roots in the presence of these agrochemicals. Now, if you look at the lab experiments with direct contact, we found that the strains were sensitive to 16% of the agrochemicals tested. So we came up with recommendations for spray tank mixing, if you choose to do this. The PR6, PR3A blends, they can be mixed with insecticides, fertilizers, adjuvants, and biostimulants, and most herbicides. And this is because the spore germination and viability was not affected. But we suggest avoid mixing these blends with some fungicides that are listed on the slide, and also boron, and that's because of the potential fungicidal effects on spore germination. Spore germination can be reduced. And the other direct contact experiment was when trichoderma was applied as part of a seed coat recipe. In this study, the trichoderma root levels were measured at 27 days, so 1 month and 2.5 months after the trichoderma and the fungicides were applied to the seeds. And you can see, if you look at the 4 bars on the left-hand side of each graph, that the fungicide treatments had similar or enhanced trichoderma root levels compared to the control, and the controls are the hatch bars on the right. So this was an important positive result, because this shows that trichoderma can be safely applied with these fungicides at seed coating. Now, in the indirect contact experiments in the greenhouse, we found that trichoderma was tolerant to all of the agrochemicals tested. So this included the soil prep herbicides that were sprayed before sowing, the soil drench fungicides applied either just before or just after sowing, and the soil drench herbicides at sowing. So this tolerance may have been because the growing media provided a buffer between the trichoderma and the agrochemicals, even when the agrochemicals were moving through the soil. And sometimes we found the trichoderma root levels were enhanced, that was shown in the previous graph, and possibly the trichoderma was able to dominate the other resident fungal species. Trichoderma was also tolerant to aerially applied herbicides, fungicides, and boron sprays, and this may be due to there being a sufficient distance between the trichoderma and the roots and the applied agrochemicals. So overall, this gave us confidence that the selected trichoderma strains are compatible with most of the forestry agrochemicals at standard rates, particularly when they are applied to the growing media. We also did a small amount of work which suggested that these trichodermas were tolerant to agrochemicals at higher rates when they were applied to the growing media. Now, in this section, we are looking at the impact of trichoderma on tree growth and disease suppression in plantations. So we've set up validation trials to test the PR6 and the PR3A blends. These blends were applied to the seed in the nursery, and we are comparing these to the untreated control. There were 8 trials set up in 2018, 2 in Gisborne, Northland, Bay of Plenty, and Nelson. They were large scale with a robust design, so there were many trees per plot and a high level of replication. A new series of validation trials has recently been set up in the South Island, looking at whether these trichoderma blends will perform well in cooler and sometimes drier environments. So they were established in winter 2020 and 2021, with 1 trial in Nelson, 2 trials in Canterbury, and 3 in Otago. Again, they were large scale with a large number of trees per plot and replications. So if we look at the results of the 2018 trials, first in the two graphs on the left-hand side, in year 1 and year 2, the average height was at least 6% higher in the trichoderma treatments compared to the control. And in the 4 trials where we were able to measure trunk diameter, this is the graph on the right, the diameter was 9 or 10% greater in the trichoderma treatments. So trichoderma has given a significant boost to early plantation growth, and we're looking to measure these trials again in year 4, which is next winter. So we're starting to measure foliar disease. It's mainly Dothostroma at this stage in these trials. So this is an example of one of the Kaingaroa trials where there was a 45% reduction in disease severity, and that was measured last November. So this is a promising result so far, but we need more measurements over time. So for the ongoing or future research, we want to continue to quantify the effects of trichoderma on growth and disease suppression in nurseries and plantation trials. We continue to study whether these strains can be applied to uninoculated forests to increase growth and health, but we need to find a practical way of applying the trichoderma. Can trichoderma be used to boost poor or new production soils in nurseries? So we're starting to get good results with this and getting good nursery feedback as well. We need to find out or continue to find out how trichoderma is persisting in plantation trees, how long it's lasting. And we have projects looking at whether trichoderma can control canker in cypresses and Swiss needlecast in Douglas fir. We'd like to start to do new research into whether trichoderma can be used to improve stand uniformity and to show whether these trichodermas are contributing to increased needle nutrition in the validation trials. So in conclusion, there are early indications that trichoderma inoculated plantation trees have more growth and less foliar diseases than the untreated trees. And in addition, we've found these trichoderma blends are beneficial in other forestry species as well. So these trichoderma blends appear to be a promising, effective growth and biocontrol tool for New Zealand forestry. We've found they can be successfully integrated into most production systems that use agrochemicals and other products. And there has been good uptake in nurseries this winter, with inoculum for 60 million seeds being sent out. And this is all part of the process to help evaluate a business case for the commercialisation of a trichoderma blend for New Zealand forestry. Now, I get a huge amount of help and support from industry partners. So this is a big thank you to the companies that are listed on this slide. So thank you for taking the time to listen to my talk.